Welcome everybody back. Welcome everybody back from the break for our final session of the day, which is session 3, Machine Learning in Computational Chemical and Material Sciences. And Professor John Straub from the Chemistry Department at Boston University will be introducing session 3. John's research interests include dynamics, reactivity, and energy flow in molecular systems, multi-scale models, and enhanced sampling methods for the simulation of materials in biomolecular systems, molecular self-assembly in complex liquids and membranes, and probing the principles of protein aggregation and amyloid disease. John, thank you.
Hey, thank you, Emily. It's my real pleasure to introduce our third session in the symposium today that will focus on machine learning in computational chemical and material science. So computational methods are able to resolve chemical physical phenomena from electronic structure to atomic and continuum levels. However, this requires high performance computing and long compute times, as Frank demonstrated so well in his excellent presentation. Integrating machine learning and data science approaches with computational methods holds the potential, many feel, to enable more effective and efficient chemical and materials development. And in this session, the promises and the challenges of machine learning applications in computational chemical and material science will be discussed by 3 leaders in the field and through the panel discussion. So let me first thank BU faculty organizers supporting the development of this session, Emily Ryan, Associate Professor in Mechanical Engineering, but also David Coker, our Director of the Boston University Center for Computational Science and a Professor of Chemistry, and Sahar Sharifzadeh, Assistant Professor in Electrical and Computer Engineering at Boston University, and finally Qiang Sui, or QC, Professor of Chemistry at Boston University. So let me welcome our 3 presenters today and introduce our panel moderators. So first, our speakers will enjoy talks from Pratyush Tiwary, who is currently assistant professor in the Department of Chemistry and Biochemistry at the University of Maryland in College Park. Michele Chiriotti is associate professor at the Institute of Materials at the Swiss Federal Institute of Technology in Lausanne. And finally, Yushan Lin is associate professor of chemistry at Tufts University. So this session's moderator is my colleague, QC. His research interests include protein allostery and dynamics, effective quantum and classical, mixed quantum classical methods for simulating large biomolecular systems, dynamics and reactions at solid liquid interfaces, as well as multi-scale computational methods development and models development and applications. So finally, let me remind you of a few housekeeping rules. Today's session is being recorded and will be posted at the Hariri Institute's YouTube channel this week. And note that all questions should be held until after the talks and reserved for the panel discussion, and the members of the audience are encouraged to participate using the Q&A feature. And with that, I'll turn things over to our first speaker, Pratyush Tiwary.
Okay, so I hope my screen is visible. And let's go to... Okay, so the atoms are moving, I imagine. Great, so my name is Pratyush Tiwary. I'm an assistant professor at University of Maryland. And I'm very, very excited to be here with you today virtually, even though I would have really loved to be in Boston in person, to tell you about some of the fun my lab has been having at Maryland in the last 4 years since we started here, building up on the work, of course, I did over the years with my mentors in the past. So, and in the last session, we had a discussion about how to train students who can do both chemistry and machine learning. So I'm really proud of showing you the composition of my group which at last count has students from 7 different departments, which is one of the things we have been able to have at the University of Maryland, including my newest student who's a mathematician, one of my newest students. So we have a great time discussing and being able to do the type of things I will show you today. And these are the organizations which have supported us through either financial support or through supercomputing time. We are happy to use up GPUs or CPUs wherever we can get hold of them. So the main motivation of my lab when we started 4 years ago was this, and this is going to be a pop quiz for the audience, okay? This is a post-lunch session. I want everyone to be awake, and Zoom makes it so hard to see who is dozing off, so I'm gonna give you a pop quiz, especially since Hariri Institute gave me the option of giving a pop quiz. So here is a book chapter, and your question is whether this comes from the book on deep learning by Goodfellow, or if it comes from Mark Tuckerman's book on StatMech. And I'm going to give you 15 seconds, and then the poll will close.
Apparently, panelists can't vote.
Yeah, you guys are too knowledgeable. Okay, 5 seconds remain. 3, 2, 1. Okay, we are done. Can we see the results? As you will notice, the majority got it wrong. It is not from Mark Tuckerman's book on StatMech. I should take a screenshot of this because, yes, this is good. So as you can see, a lot of you thought it is from Mark Tuckerman's book on statistical mechanics, but that is indeed not true. This one comes from deep learning book by Goodfellow. And if you like StatMech as much as John or QC do, you would have realized no self-respecting book on StatMech would talk about partition function in chapter 18, right? But it is confusing. If you look deeper, you will see the name of Jarzynski, who is my colleague here at Maryland, and it's confusing as to where it comes from. And which was really our inspiration, that both these fields have been tackling kind of the same problem. In statistical mechanics, we know it as finding the partition function. In machine learning, you call it the normalization constant, whatever you want to call it. But it is this one quantity that contains all thermodynamic information about a system. If you can find out statistical mechanics, if you can find out the partition function, you can play a variety of tricks with it. You can differentiate it. You can take its log and then differentiate it, or do whatever you want to do to get all sorts of wonderful thermodynamic information. The problem, though, it is very, very hard to find. So our inspiration was both these fields are kind of tackling the same problem. Could we see what synergies are there to be explored? What's harder, though, is we don't want to just calculate thermodynamic properties. We also want to calculate kinetic properties or dynamic properties. Why do we want to do these dynamic properties? By which I really mean we want to do molecular dynamics. We can study really, really important and practical problems with this. So I will show you some applications that I don't know how much time I will have to left to go over these, but these are all problems that my lab is tackling right now. These are not problems that we one day might be able to tackle. So for example, this is a drug called Gleevec. It's a blockbuster drug for treating certain forms of leukemia. And when it came out in 2001, it made it to the cover of Time magazine, and it was a super hit drug. Only 13 years later, it was featured on the cover of Newsweek in context of other drugs with the title Getting Cancer Wrong. So what would happen is that this drug goes and blocks a particular protein called a kinase, and patients would undergo mutations. The drug would become ineffective if there is one single point mutations, even so far out from the binding pocket. So not all of these mutations would affect the drug, but some very specific ones would affect it. So why would such a mutation so far out from the binding pocket affect the drug? This is something which is very, very hard to explain and continues to be very hard to explain. You cannot just explain it by looking at the structure of the drug bound to the protein. It's so far out, it's almost 20 angstroms or more away. So we would like to be able to simulate the protein and really see what's going on. Maybe the drug goes through this part of the protein when it's associating or dissociating. What is the reason for this to happen? So this is one problem we are interested in. The other problem we are interested in comes from material science, which is kind of like Frank. I am also trained as an engineer, specifically a metallurgical engineer. So problems such as how do thin films grow, or how do dislocations nucleate when you compress a pillar, or a problem which have continued to be of immense importance over the last centuries is nucleation. In this case, you're looking at atoms of urea nucleating into different polymorphs. This is work done by my students, Connor and Sunting. And we want to study which polymorph forms, when, what are their relative free energies. And again, these are all very important problems. And the third one is how do proteins fold and misfold. I especially want to highlight misfold because to some extent, and this would probably come up in the panel discussion, AlphaFold has already solved perhaps the folding problem. But often biology is not just about the ground state, it's also about the excited states when the protein adopts a conformation, which is not the ground state, yet such a conformation, which can depend tremendously on small changes in the sequence. For this protein over here, you make 1-point mutation, 2-point mutations, 3-point mutation, the whole energy landscape shifts. And we want to be able to predict how this shifts, what are the barriers for converting between them. So for all of this, we would like to use molecular dynamics, which is as simple as solving Newton's law of motion. This is apparently Newton with his pandemic hair from early 17th century. So it's simple in principle. You just solve F is equal to ma, but it's really, really slow, right? If you're solving equations with femtosecond with supercomputers or with GPUs or with Wall Street money, you can maybe get up to a millisecond if you run it long enough. But for problems such as nucleation of a crystal or protein ligand unbinding, it's not sufficient. So on one hand, it produces too little data because you cannot study anything of interest. On the other hand, it produces too much data. If you don't believe me, let me tell you about the number of times my students have filled up 10 terabyte data on the hard disk and I get angry email from supercomputer administrators. It's easy to produce lots and lots of data, which means nothing. So we have a regime where AI should be usable, but it's not really clear how because we don't have enough training data. And furthermore, we have been able to study bigger and bigger systems with complicated supercomputers by just breaking the system into different chunks. We can parallelize large-scale computing. We cannot really parallelize long time-scale computing because time is inherently sequential. And this plot that I'm showing you here, this is a classical force field. Once we go to polarizable force field or Born-Oppenheimer or even fully quantum dynamics, this problem will become even more important. So the approach that we are taking, or the question we are asking, is it really true that time is inherently sequential? Or could we get away by knowing just a little bit about the past state of a system to make accurate predictions about the future? This is where AI comes in. People have tried over the years to use various models of AI, such as bidirectional autoencoders. Well, if it can tell a dog from a cat, or if you can use it on the internet to mix faces and even recognize handwritten digits from each other, why shouldn't you be able to use these latent variables as proxy for the predictive degrees of freedom in a simulation? What do I mean by these predictive degrees of freedom? These are the few degrees of freedom for a complicated high-dimensional system which captures everything. So when you are doing it on a face, on a collection of faces, hopefully this latent variable corresponds to something like hair or nose or color of the face in handwritten digits. I don't know what it corresponds to, but the idea is instead of clustering in the high dimensional space, if you cluster in the low dimensional space, you can generate semantically meaningful samples, as we saw in the last session. So people have been inspired over the decades to use these latent variables to do something called enhanced sampling, to use it as a proxy for what in chemistry we know as the reaction coordinate. And then you can play a variety of tricks developed over the years to basically speed up the sampling along this reaction coordinate. So here I am citing some of the people who have worked on this problem in the recent years, but the approach, the motivation to do this even goes before this, because people have tried to use principal component analysis as a proxy for this reaction coordinate even way before this. But the problem is it does not always work. There is motivation that it should work. For example, in context of AI, these were two seminal papers that came out in 2014, and this, which probably is more famous, the top one and the bottom one in 2017, which showed that if you're doing a deep neural network, if you're performing a deep neural network on a physical system, the layers of the deep neural network are automatically learning. In this top paper, David Chop and Pankaj Mehta show that they are learning something which is akin to the group block spin structure. So when you go to the deepest layer, you have learned something which is similar to the magnetic moment. So, but this is all clustering. There is no guarantee that this should function as the reaction coordinate for physical chemistry. So this is what brings us to our approach, which is to go slightly deeper than autoencoders. We look at something called information bottleneck, which was pioneered by Bill Bialek. And originally the idea goes back to Shannon. What Bill Bialek says in the information bottleneck concept is that a good model of a system should have two properties. It should be as predictive as possible. So you have a funnel which opens up, while at the same time, it should be as compressive as possible. So it should be able to distinguish the signal from the noise. It should not try to remember everything in the data. So this information bottleneck can be written down as a neat mathematical principle. In order to learn an information bottleneck, you minimize the mutual information between the bottleneck and the input, and you maximize the mutual information between the bottleneck and the output. In our case, we take the input as an MD trajectory, as a molecular dynamics trajectory at a certain time. And as the output, we take the trajectory certain time into the future. So notice this is already different from variational autoencoder. Traditionally, variational autoencoders don't do this. They take the input as the same as the output. And this Lagrange multiplier over here, or a constraint or a hyperparameter, is set to be equal to 1. So what we are doing is something similar to a time-lag variational autoencoder, but not quite. And we can get into details if there are questions. But so in principle, the problem is solved. If we have a molecular dynamics trajectory for training, we could pass it through such a network. We could approximate both of these funnels through deep neural networks and learn the reaction coordinate. We can learn the information bottleneck that contains everything that is to be known about the input in order to be able to predict the output. So in one hand, you have compressed the molecular dynamics trajectory into a low degree of freedom so that everything else that you don't know is per construction, not predictive. It does not carry any predictive power. And in this very recent paper in Journal of Chemical Physics, we showed that indeed this approximates what in chemistry is known as the committer, which is known to be the perfect reaction coordinate. And you can go and look up this paper where we show it follows certain nice properties such as this committer distribution expected of good reaction coordinates. So now I can show you the algorithm, the way it works. You have a trajectory which is trapped in one basin, or it does not really go back and forth because the time scales are so limited. You take this trajectory, you pass it through an encoder-decoder framework. Notice here I'm showing you a linear encoder, which we kept. This is, you can call it a shallow network, so that the network is interpretable. Now we have generalized it in many ways. To take this bottleneck, since you know it is a committer, you can, as we show here in this paper, you can trust it. You use it in one of these many methods that have been developed over the decades. These are called enhanced sampling methods, where once you know this information bottleneck, you can play tricks with the energy landscape of the system. You can go and fill up basins where the system would get trapped. So it's encouraged to go into other side. And then the next time you run this simulation, your system is no longer trapped. It is going back and forth. Don't stop at this point. Take this superior trajectory, go and learn a better information bottleneck, and keep iterating. This is also in a strange way similar to what Connor and others were describing in the morning session, where you can think of molecular dynamics as doing a complicated experiment. You don't stop with the first round of experiment. You use it to learn, design better experiments. This does not solve all the problems. Oh, by the way, and in order to feed these input order parameters into the molecular, into the bottleneck structure, we have yet another pre-processing method which simplifies life. I don't have time to go into it. But the point I want to emphasize in a few seconds is this framework of just iterating between machine learning and molecular dynamics works, but often does not work. And the problem is that deep learning is, in the end, as we heard from many speakers, a stochastic gradient optimization problem. So if it works well, for example, here you upload a picture, it generates a text. These are examples when it works wonderfully well. Often it fails. Surfboards on a beach, no, the text generated was a bunch of bananas are on a table. Cat looking at pigeons, no, group of people standing around a horse. Bird on a beach, no, small plane flying in the sky. So if you just take a deep learning network on its face value, it can mislead you. And that brings us to yet another connection with StatMech, which is if we have two solutions from machine learning, we pick the one with the highest spectral gap. Learning this spectral gap is hard. So we have to use this concept of a maximum caliber, maximum path entropy model to quickly learn the spectral gap, but we can optimize all of this and put it in one routine where anytime we have many solutions from AI or machine learning, we pick up the one with the highest spectral gap and use it for the next round of molecular dynamics. So as you can see here, the same value of the loss function of the information bottleneck function can correspond to different spectral gaps. And they have an overall 1:2 negative correlation, but in the middle, it can mislead. So this work, which was featured in JCP recently, showed how this framework can go and, at least in molecular simulations, help with this spurious min-ma problem.
Ding.
Okay, so we have more methods, and I could again, I would have to give you a much longer talk to show what's going on. We also use recurrent neural networks to make sense of data once we have high-dimensional data. But I want to just show you one application from RAVE, which is the dissociation of the small ligand from RNA. So a year ago, I would not have casually spoken about RNA because not many people knew about it, but thanks to coronavirus, I wouldn't thank it, but a lot of us have suddenly realized that RNAs exist and they are important. So my collaborators at NCI want to design drugs that can bind to RNA, but they are so flexible that it's very hard to figure out what goes on. So if you do MD on these molecular dynamics, very long molecular dynamics, you can see that they reproduce the experimental flexibility, but the ligand stays trapped, nothing happens. So now if we do RAVE simulations on this, which is iterating between machine learning and molecular dynamics, we can actually get the ligand to dissociate. And for these two different ligands, we can see which is the preferred pathway. So what we find is that this cognate ligand, the one in red, goes through the top, while the synthetic ligand goes through the bottom. Why is this important? Because this again goes back to that Time magazine cover that I was showing you in the beginning, that we can now show you directly which mutations will make the ligand ineffective. So our collaborators went and made these mutations, and they indeed found our predictions to be true. So these are 4 sets of predictions, and they were quite surprised by this. So this was very exciting for us. And similarly, we have similar results on the kinase, where we can go and explain, we can calculate the residence time of the drug using all atom simulations, using variety of statistical mechanics ideas. All of them, the key input here is the reaction coordinate, which we are learning by iterating between machine learning and molecular dynamics. And finally, I will show you one slide, which is how a small protein folds and unfolds. This is barely a protein, it's just, but it can form a alpha helix and beta hairpin. With that, I think I can conclude. I showed you that with the help of AI, you can go and accelerate molecular dynamics, but AI is, even though it has intelligent in the name, it can be a bit stupid at times. It can get stuck in local minima. Coupling with StatMech is one way to solve this problem. We have a lot of methods. All of them are on GitHub. You can go and play with them. And I think it's a very exciting time to be learning chemistry or biochemistry along with AI. And because it's a wonderful time to be doing that. And we have openings for grad students and postdocs. With that, thank you very much.
Thank you very much, Pratyush, for a really excellent talk. So think of your questions for the panel discussion later. And now the screen is yours, Michele. I think you're muted.
So thank you very much for inviting me here. I'm very excited to be speaking, even if unfortunately only virtually. And I'm going to talk about how machine learning is really changing the face of simulations at the atomic scale. And in doing so, I'm going to show you a lot of examples and a lot of applications. And before I start, I want to make sure to have the opportunity of giving a shout out to my group who has been actually doing most of the work. So to give a little bit of context, actually, my background and my interests are not too different from those of Pratyush. Actually, we have both been students in the group of Michele Parrinello. And, you know, what you're trying to do, what our dream, I think, both of us is really being able to achieve a quantitative, predictive description of the emergent physics of complex systems. Proteins, but also phase transitions in materials. And when you try to do this, you're really up for a very, very hard challenge, because on one axis, you need to converge the description of the energetics and the properties of your system, meaning that if you don't want to parameterize your models on experiments, you have to resort to a solution, usually approximate, of the quantum mechanical electronic structure theory problem. On the other axis, however, you need to worry about sampling fluctuations, rare events, which is basically what Pratyush works, does for a living. And the combination of these two means that this is essentially an unsolvable challenge, and it was unsolvable until machine learning came around. And essentially, you know, it's nothing really magic, but it does give you the opportunity of essentially just fitting a small number of reference first principle calculations so that you can then recompute the properties and the stability of similar structures at a cost which is only marginally greater than that of a force field, which means that these large-scale, long-time simulations that you need in order to describe phase transitions, chemical reactions, and all this kind of interesting physical phenomena is now becoming really possible. So at a certain level, the way this works is really nothing particularly fancy. What you need to do at a certain point, and you probably have already heard quite a bit of this, is training a model. And training a model means that you define, first of all, a functional form that links an appropriate description of an atomic structure A and tries to predict the properties computed for this structure. And then you fiddle around with the parameters of this model until you obtain a small error in reproducing the properties of this structure A. And you know, there is a whole lot of different families of models of different complexity. And you really can span from linear regression, which actually can work great, to the fanciest deep learning models. And one message that I want to give is that not necessarily a more complex model is a better model. And this is because, yes, a more complex model gives you, in principle, the possibility of being more accurate in reproducing these structure property relations, but typically requires a huge amount of data to do so. And if you want to predict accurate target properties, these reference calculations don't come for free. You need to minimize the number of reference calculations that you need to do. And in many cases, actually a simpler model, even linear regression, but perhaps something intermediate like kernel methods, can actually give you better performance. Now, besides the choice of the regression model, or also the choice of other machine learning schemes that can be useful for other tasks, clustering, dimensionality reduction, and so on and so forth, a crucial ingredient when you want to apply these methods to atomistic simulations is the choice of a representation. So how do you process the Cartesian coordinates of the atoms in your molecule or your material so that they can be suitably used as inputs for a machine learning technique? And perhaps the best way of realizing how important this is just to look at how many different frameworks have been proposed over the past decade to perform this mapping between structure and features that are then used as inputs of a machine learning scheme. And even though there are really several dozens of frameworks available that we try here to sort of organize in some kind of phylogenetic tree, we are increasingly becoming aware of the fact that the vast majority of these representations, of these featureizations, are actually very closely related with one another. And the reason why, staying in this sort of biological metaphor, the reason why they are so similar is that they have undergone a process of convergent evolution to fulfill the same set of requirements. So typically when you map structures to feature spaces for machine learning, you want, and this is actually not such a trivial requirement, you want this mapping to be complete. So you want that if you have two structures that are fundamentally different from each other, they should be mapped to different points in feature space. And this is because otherwise, no fancy machine learning scheme you apply after this representation step will be able to predict correctly different properties for these two structures. At the same time, in order to achieve a compact and effective description of your system, you want your feature mapping to fulfill some symmetry requirements. So you want that if you have two structures that only differ because of a trivial symmetry operation, a rigid translation or a rotation, they should be mapped to the same point in feature space. And again, this may seem like a trivial requirement, but Cartesian coordinates don't fulfill this. Then you want the mapping to be smooth, because usually the mapping between structure and properties is smooth, and so you want this intermediate step to preserve this property. And finally, and this is actually a very important requirement in order to be able to build models that you can transfer from simple systems to more and more complicated ones, you usually want a representation that works by decomposing the description of your structure into a sum of descriptors of atom-centered environments. So essentially, rather than describing your entire structure with a single vector of numbers, you describe each atom and its atomic neighborhood with a separate vector of numbers, and you will build a model that predicts the properties of the whole as a sum, as a combination of the properties of its parts. So, as I anticipated, over the past couple of years, we have been working towards some kind of a general theory of representations, and I don't really have the time to go into the details, but I just want to give you a flavor of what is the idea and how it works. And what is important is that pretty much all of the representations that I showed on that kind of big family tree can be seen as projections of the objects that arise from this construction onto a suitable discrete basis. So starting from the Cartesian coordinates of the atoms, the first thing that you want to do is make sure that your representation doesn't depend on the order by which you list the atoms. Again, a trivial symmetry that is not fulfilled by Cartesian coordinates. And you can do this by associating each structure to a density, which is built by summing local functions centered on the atoms. And then you can build a hierarchy of representations that correspond to endpoint correlation functions of this atom density. And if, like me, you like statistical mechanics, these are basically the same objects that are used in the statistical theory of disordered systems. And also something that I find very pleasing is that if you build a linear regression model, so the simplest possible regression model on top of these features, you recover the body ordered expansion of potentials. So basically you derive properties as a sum of a function of the distances between atoms plus terms that depend on the relative correlations of 3 atoms, 4 atoms, and so on and so forth. And this framework is actually very flexible, and you can extend it also to be able to machine learn properties that are tensorial in nature. So for instance, vectors or polarizability matrices and so on and so forth. And you can also extend it to incorporate into this same atom-centered framework a description of long-range effects such as electrostatics. So let me now give you a few examples of what can be done using this kind of framework. So a first thing that you can do is compute very precisely and without any input from experiments the finite temperature thermodynamics of materials. So here, for instance, we can compute the melting point of water, including the effects due to the quantum nature of the nuclei. For instance, we can get the difference between the melting point of light and heavy water. And we can even compute and look at the energy scale. This is minuscule. It's a difference in free energy of a few millielectron volt per molecule. We can compute the difference in free energy between the hexagonal and the cubic form of ice, essentially explaining why snow flakes are hexagonal rather than cubic. You can also use it, and I mean, this is one of the big advantages in general of first principle calculations. You can use them to study matter in conditions that are hard to probe experimentally in a direct fashion. And for instance, you can apply machine learning to the study of matter in extreme conditions, such as in the core of giant planets. And you can use it to reconstruct the phase diagram of a binary system, in this case, gallium arsenide. And what I want to stress here is that the error between the experimental and the computed phase diagram is largely due to the uncertainty in the reference calculations, in the approximation to the electronic structure theory that we used to train the machine learning model, more than due to the machine learning model itself. And besides this problem of accuracy, there are other limitations in the current application of machine learning to atomistic modeling. And one of these, I think, is very well explained by this other application, and this is the study of a solvated electron in water. And since in order to study the properties of this solvated electron, it was necessary to compute the electron density associated with this excess electron. After having run a molecular dynamics calculation based on a machine learning potential, we had to go back and run density functional theory to get access to the electron density. And so one of the things that have been keeping myself and my group busy is actually building machine learning models for properties with a complicated algebraic structure that goes beyond the simple scalar nature of potentials. So for instance, you can learn polarizabilities, you can learn electron density, and there is an archive paper that came out yesterday in which we showed that this can also be applied to the condensed phase, and you can learn electronics excitations in terms of the single particle density of states. And overall, the combination of machine learning potentials and these models that allow you to predict the dipole moment or the polarizability means that now you can have access to what I call integrated machine learning models that make it possible to really predict essentially from first principles, the properties, also the functional properties of materials, for instance, infrared or Raman spectra. You can compute the heat capacities of materials, including also the contribution that comes from electronic excitations. And you can do this scaling up your simulations to large scale and long time frames so that you can really study the behavior of matter in a regime that is not available to empirical force field models and is not accessible to explicit electronic structure calculations. So all of these is available in a sort of a stack of open source code that is available for everyone to get. Just need to go on our GitHub page. And to sort of wrap up, my main take-home message is that data-driven techniques are really transforming the modeling of matter at the atomic scale by making accurate potentials affordable and by getting models that are more flexible than the physics-based empirical force fields are. But also physics and physical principles are making their way into data-driven techniques on one side by providing training targets, but also by providing principles, symmetries, locality, and conservation laws that make machine learning models more efficient. And to conclude, I really believe that the interface between data-driven and physics-based modeling is where the next generation of atomic scale modeling techniques will arise, getting sort of the best of these two worlds. So thank you for your attention.
Well, thank you, Michele, for a terrific presentation. And now is our final speaker of the symposium, Yushan Lin. Yushan, take it away.
Hi, everyone. Today, I'd like to share with you some recent effort my group has been doing on integrating molecular dynamic simulation results and machine learning to predict structural ensembles of cyclic peptides. So I first would like to thank my hardworking group, and for this talk, especially Dr. Miao, a graduate student Tiffany, and undergraduate student Mark. Why are we interested in cyclic peptides? So one of the reasons is for their ability to modulate protein-protein interactions. So in each of these images shown below, red is a huge protein, blue is another huge protein. They come into contact, trigger some conformational changes, signal gets processed. So these protein-protein interactions are prevalent in biology, and in many diseases, specific protein-protein interactions are overexpressed. Therefore, there's a great interest in being able to modulate these protein-protein interactions. So how do we go about doing it, and then how do cyclic peptides come in play? So in the figure shown below, we can see the three main categories of therapeutics currently available. So on the bottom left, we have small molecules, which typically obey Lipinski's rule of 5. So the molecular weight is generally smaller than 500 Dalton. And then because of the small size, they typically have very good pharmacokinetic properties. However, also because of the small size, they are not able to interfere with something as large as protein-protein interactions. So if you are interested in modulating protein-protein interactions, on the other hand, you need something more substantial, such as biologics, which are antibiotics and other proteins. So these large proteins are able to target protein surfaces with high affinity and selectivity. However, they usually have very poor pharmacokinetic properties, meaning that you are able to take the drugs by mouth, for example. Now situated in between are cyclic peptides and other macrocycles. They are considered beyond rule of 5. So their molecular weight is larger than 500 Dalton, but however, they can actually have good pharmacokinetic properties. They are also able to target protein surfaces with high affinity and selectivity. So the hope is, can cyclic peptides combine the best attributes of the two worlds? And thanks to a lot of structural biologists, we actually have information on a number of protein-protein complexes. So if you go to the protein data bank and look at the protein-protein interactions, people have characterized more than 50% of the time, what you're going to see is that the interface residues adopt these non-regular secondary structures. So if I zoom in, it almost looks like the blue protein is forming some kind of loop-like structure and target and glue to the gray target surface. So these are now what people term hot loops. So these are loopy-like structures that contribute a lot to the binding affinity. So therefore, if I want to inhibit the protein-protein interactions, what I can do is, well, I can design something that specifically mimics this hot loop, right? And I know it has a very high binding affinity for the gray target surface. In a naive mind, what's closest to a loop is a circle. So what you can do is, for example, I know I need serine glutamate to be arranged in this specific conformation. So I can synthesize these 4 amino acids and add a couple more, do a head-to-tail cyclization, and give you a cyclic peptide. But then the key thing is, I need this green portion, the serine glutamate, to point exactly in that way I see at the protein-protein interface. How can I actually design the linker then to best stabilize the desired loop structure? I will be able to achieve this if I actually know how sequence controls the structure of the cyclic peptide, right? So if I can predict cyclic peptide structure, then I can say, hey, when I use this linker, this cyclic peptide will orients the side chain of SESE pretty similar to what I see in the active form at the protein-protein interface. Then presumably this will be a very good binder. If I don't design my linker correctly, then now I have a cyclic peptide that still has SESE, but they are pointing in some directions. They are not compatible with binding of the gray surface. Then this will likely not be a great candidate. One of the main challenges in designing cyclic peptides is that it's currently very difficult to establish the sequence structure relationships for these molecules. Why is that? It turns out that cyclic peptides tend to form multiple conformations in solution. The implication is this makes it very difficult to use solution NMR spectroscopy to characterize their structures, because solution NMR spectroscopy typically assume that you have one single conformation in solution and try to derive a structural model for it based on the NMR signals. You know that I'm a computational chemist, so you probably can guess what I'm about to say. Why don't we use, for example, molecular dynamic simulations then to characterize the structure ensembles of cyclic peptides? Then I can say, hey, I designed linker sequence number 1, the resulting cyclic peptides have multiple conformations. In my simulation, I can tell you what the populations are. And in this case, only 2% of the time orient the SE residues correctly, probably not gonna be a great drug. And then in sequence number 2, ooh, like this population now goes up to 70%, so this will be a better candidate than the first sequence, and so on and so forth. In molecular dynamic simulations, what we actually have found is small cyclic peptides, they do not like to switch conformation because of the ring restraint. However, we also found that when they do switch conformations, it typically involves 2 dihedral angles simultaneously. So in general, it will be the phi and psi of the same residue switching at the same time, or the psi and the phi of the next residue switching at the same time. So by targeting these essential transitional motions of cyclic peptides using metadynamic simulation, we are able to obtain converged structural ensembles in 100 nanoseconds of bias exchange metadynamic simulation. However, this kind of simulation is still very time consuming and computationally expensive, and it is not feasible for screening a large number of sequences. Is it possible that we can actually replace the need of running these time consuming MD simulations with a machine learning model? So today, what I want to share with you is how we are thinking of framing this problem that when you give me an input, which is a cyclic peptide sequence, I want to be able to provide you a predictive structure ensemble for the solution structures, as you would get if you run the time-consuming MD simulations. There are 3 aspects I want to touch on today. Before that, in the example I'm using in this talk, we will focus on cyclic pentapeptide, and the amino acid will be one of these 15 kinds, and the lowercase letters represent D amino acids. The 3 aspects I want to touch on today are 1, how do we represent cyclic peptide structure ensembles? 2, how do we actually formulate this problem? 3, how do we design the training data set? Currently, how we represent cyclic peptide structure ensembles is by using the structural digits derived from cyclic G5. So glycine is the smallest and most flexible amino acid. So we run the simulation of cyclic G5, and then we get the Raman-Chandra plot, which is the density distribution along the backbone phi and psi dihedrals. Then we decompose this density plot into 10 different areas. So we call each of them a structural digit. Then if you give me a cyclic peptide structure, then I can look at the phi psi angles of each residue, and then based on where they land in the map, I can assign each of the amino acid a structural digit. So this enables us to describe a cyclic peptide structure ensemble using these structural digits, and then we can also report the population. How do we formulate the problem? So what we want to get out of is a population of a specific cyclic peptide amino acid sequence adopting a specific set of structural digits, S1, S2, S3, S4, S5, at amino acid X1, X2, X3, X4, X5. In a chemist's mind, the population is gotta be related to the energy of this conformation. So in the numerator is the Boltzmann factor. You can see the energy shows up here in the exponent, and the Q is a partition function for normalization. If we today only consider 1, 2, and 1, 3 interactions for the energy, then what we end up with for cyclic pentapeptide is I will have 5 1, 2 interaction terms, and I have 5 also 1, 3 interaction terms. When we take the natural log of the population, then it will be related to the 10 interaction terms I just mentioned, plus a normalization factor term. This enables us to write the problem into a metric format. So this will be a series of the populations in your training set you want to fit to. Then this is a long array of weights. And then the coefficient matrix, basically each row picks out which 11 weights you need to describe a specific population for a specific sequence and structural digit. Then we determine these weights by minimizing the following loss function with respect to weights, which is the difference between the fitted population and the observed population. given that this is how we formulate a problem, how do we design a training data set? So currently what we have done is we generate sort of random sequences. We want to cover all the possible X1, X2, X3 sequence patterns. And we try to do so by using as small a set of sequences as possible. So here is the result of the training. So then the Pearson coefficient between the observed population and the fitted population is 0.996 with a weighted error of 0.632. When we test the model's performance on 50 new random sequences, then we still have very, very good Pearson coefficient between the observed population and the predicted population of 0.960 with a weighted error of about 9.6. So overall, I'm pretty happy with the performance of our current model. And then I think this could be a very useful way to solve this challenge of establishing sequence structure relationships for cyclic peptides to enable its structural design for therapeutic purpose. Since I have about 4 minutes left, I want to also share with you some dirty little secrets I didn't tell you about, and then what my group is currently working on in regarding this project. So first, when we talk about how to represent cyclic peptide structure ensembles, we are currently using the structural digits derived from cyclic G5. If you look at this Raman change-out plot for this new cyclic peptide carefully, I don't know if you noticed, but these three residues, you notice there's a very sharp cut for its density, right? So this is a sharp boundary generated over here between the beta and pi. This is over here, and then this lambda is over here, right? So what this suggests is the map you gave me, is not a natural cut for this new cyclic peptide, right? So meaning that the structural digit map derived from cyclic G5 might not be very appropriate for other cyclic peptide sequences. Therefore, we are currently working on, instead of predicting structural digits and the populations of each structure, we would like to predict, for example, three-dimensional density profile in a common principal component space. So here is a pilot study. So these are the test sequences. On the top is the predicted three-dimensional density profile of the cyclic peptides in the principal component space. On the bottom is the observed three-dimensional density profile if you actually run the time-consuming simulations. So in general, we can see that we are able to see the general features being kept and also the differences between these cyclic peptides. Another aspect I want to touch on is how we formulate the problem. So in this case, we stick to relate population to energies, which, as Mikhail mentioned, this is, you have great interpretability, but then how about your performance, right? So then we are also thinking about giving less control of the model, right? So for example, do a neural network and then see if we can further increase the performance of the machine learning model. I also mentioned that currently how we design the training set is to cover all possible X1, X2, X3 amino acid sequence patterns. This is actually very difficult to expand when I want to, for example, use all 39 amino acids of D and L amino acids plus glycine. If I want to expand going from 15 to 39 amino acid, I will actually need 11,000 training sequences. So given my lab's computational resources, this will likely take a year or two to complete. How are we going to solve this problem then? So we are currently working on representing amino acids using their physical chemical properties, for example, the hydrophobicity, hydrophilicity, polarity, polarizability, and so on and so forth. So then our model will not be using a one-hot representation, but it will be trying to relate the physical chemical property of amino acid at each position to their structural preferences. Today, I also focus on how we can go from sequence to structure by integrating molecular dynamic simulation results and machine learning. How about properties, right? So the experimentalists measure a lot of properties for different peptides. How can we actually predict those? It is my gut feeling that it will be pretty difficult if we want to go straight from sequence to a property like drug likeness. what I kind of foresee happen is that we can actually use structure as a springboard. So if we can relate sequence to the structure information can inform the model to be able to predict chemical or physical properties of the molecules. So hopefully today I share with you how my lab has been thinking about using computational chemistry to help design cyclic peptides. And once again, I like to thank my students and also the funding agency. And thank you for a great symposium, and I look forward to our discussion.
Well, great, Yushan, and of course also Michele and Pratyush for presenting such beautiful talks that illustrated the power of machine learning in modern chemical and material science applications. So now I'd like to welcome questions from the audience. Again, please just type your questions into the Q&A window, and I'll be happy to deliver them to the speakers. So it looks like, not surprisingly, my colleague, Professor David Coker already put some questions in there. And so I think is for you, Pratyush, but others feel free to chime in as well. So David's question is, once you learn a reaction coordinate with the data from a short simulation, then iterate as you describe. Is it clear this process will converge reliably? Suppose the first simulation was stuck in the wrong place. Why will it escape reliably to a good place as you iterate? I'm sure you thought a lot about this.
This is a great question. I will start with answering, but I would, of course, absolutely love to hear Michele and Yushan's opinion also. So to answer David's question, there are two answers. There is a good answer and there is a shortcut answer. The good answer is that I referred to a paper by Cecilia Clementi around 15 years ago called diffusion map directed molecular dynamics. The idea being that if you do a short simulation, and if you're indeed learning the dominant eigenvectors of a diffusion map, then if you go to the boundary of this eigenvector and launch more simulations, you're guaranteed to keep expanding. So you start with something that is stuck, but by going to the boundary of this isocommitter, of the first eigenvector, which approximates the isocommitter, you're basically going normal to this committer surfaces. So that's the good answer, which shows you that indeed, if you're learning the committer, you should not have to worry. By just shooting and expanding, you will get the job done, which I think is similar to one of the things Michele did back in the day, something called reconnaissance metadynamics, right? So people have tried to work on this. And in principle, these methods should work without getting stuck. In practice, however, it's hard to say. We are working on complicated problems. It's hard to say whether that is going to work or not. So in practice, what we do is, indeed, as David said, it could get stuck. So what we do is to take a bias simulation, which is coming from somewhere else, and use it as an input. And instead of just stopping with one round of bias simulation, we keep iterating on that. So now we know that the simulation has visited many places, so it's no longer stuck, but we have to make sure that the bias simulation can be reweighted, which can be a problem in many sampling methods, not in all. So, and it also leads to questions, then how do you know whether you are only in the vicinity of the path that you started with, which is a very generic question about methods, even such as transition path sampling and all. So that's my answer, and I wonder what Michele and Yushan think about this.
So for our Maynard dynamic simulations of cyclic peptides, we hope that we have identified the essential transitional motions. However, to kind of make sure that we have some sort of support that simulations have converged, we always start with initial structures that look different, right? So then we guarantee for small cyclic peptides greater than 1 point something angstrom, and then we monitor the structural ensembles you get from the two independent sets of simulations. And then if they eventually give us similar answers, then we are going to assume that at least, they converge to some degree. But then of course, it's always very difficult to prove they are doing something absolutely correctly, right? So then it's always difficult. It's always easier to verify that you didn't converge, and then promise you that I absolutely converge. But then we do start with two different initial structures and then kind of check on the convergence that way.
Yeah, I don't have much to add to this. I should only say that in general, it's actually not so hard to make your simulation go somewhere. So the problem is usually not so much getting stuck, but making sure that you converge to the right thing. You know, I started my first application of metadynamics, which is one of the techniques that also Pratyush has been using for a long time, was with ab initio metadynamics, and I was depositing hills that were 1 electron volt tall. So basically, we're throwing nuclear bombs, and the system goes somewhere. So I would say that the difficulty is not so much getting stuck, but making sure that whatever you do to make your system go away ends up converging somewhere.
So if I may add to what Michele said, I guess what you're saying, Michele, is to achieve ergodic sampling, to do back and forth, to not go somewhere else and get stuck, but come back and then go back and forth.
All right, and so this is a great discussion. So there are a few other questions already. I think 2 of them for Yushan, and so I'll start with this question from an anonymous mysterious attendee. So do you think applying the methodology to structures of higher torsional dimensionality be as effective?
So if I understand the question correctly, in our current metadynamics, we target 2-dimensional collective variable, which is phi and psi, or the sin and x phi. And then we are indeed interested in seeing that if for large side chains, you will also need to incorporate the chi angle. So that is what we are currently thinking, but if you keep increasing the dimensionality, then the space of this collective variable is going to be so large that to actually cover it well, it will take a long time. So then we are trying to gauge what's the minimal dimensionality you will need for in the dihedral space for efficient sampling. But that's a very good point.
All right, that's great. And so maybe can follow up with another question for Yushan from my colleague Adrian. If I understood correctly, say a 1,3 interaction between 2 residues in a cyclic peptide is assigned a specific landscape of structural digits. But to what extent might this landscape change based on the identity of the intervening residue and its own interactions across the ring, the size of the ring, and other compound-specific features?
That is a great question. So then I show the slides very quickly, but I want to mention that for the 1,3 interaction, for the structural digit, we do kind of care about the structure in the middle. But as you point out, we don't care about the amino acid entity identity in the middle. And then it's also for some practical fitting purpose. That is, if we care about the amino acid in the middle, then we need to see this 3 amino acid sequence many times to be able to fit this parameter accurately. Okay. And so this then for a cyclic pentapeptide, as far as I could kind of see in general, because of the size being so small, the side chain kind of tend to point to different directions. But then they certainly could be packing or interactions between residues coming in. So the 1,2, 1,3 interaction, I think will actually be a complete set for a pentapeptide, because for a pentapeptide, 1,4 will be actually equivalent to 1,3, 1,5 will be equivalent to 1,2. But then for larger cyclic peptides, there will be certainly maybe like even like 1,4, 1,5 interactions, or actually what we'd probably worry about is the multi-body interactions, right? As Adrian is alluding to. So then in this case, that's why we're also interested in moving to more like a neural network model, like that's not dictate who can interact with what, let the model figure out what's going on.
All right, so sounds exciting. So we look forward to hearing progress along that line, of course. And so maybe we can, switch to Michele for a minute. Are there methods, representative structures to check for completeness of representations and avoid related pitfalls?
So yeah, this is an excellent question. And in general, this problem of completeness is something that has been keeping myself and also good colleagues like Gavorchani very busy lately. So we have a paper from the beginning of the last year, PRL, where we basically showed that the vast majority of the representations that are being used very successfully for machine learning are not complete in the sense that you can build explicit counterexamples that essentially are clearly distinct, but are represented by exactly the same value of the features. Now, the interesting question, as it is often the case, is not so much are representations complete, but you know, how large in hand-waving sense, and how to quantify that is one of the issues. How many are the structures for which this lack of completeness shows up, and are they structures that you would encounter in a realistic atomistic simulation? So to try and address these questions, we are working to develop measures that allow you to compare different representations and check compute in a very, very vague sense and mutual information content of two different possible sets of representations, so that you can pick the one that is more information rich. But this is a very interesting and very much open question.
Yeah, absolutely. I would imagine, you know, the similar kind of questions for, you know, systematic coarse graining and things like that. Any comments from, you know, other, you know, panelists along this line? I'm sure you guys also, you know, thought quite broadly about this when you design, you know, features and such for your specific applications and problems.
Yeah, so to some extent we do. I mean, what the paper Michele mentioned is really phenomenal, and I really recommend reading it. But to some extent, we dodge the problem by taking features which are handcrafted for our problems, which is a bit of cheating, but it solves some of the problems. But if you're not taking handcrafted features, then
yeah, and in a sense, you actually don't want features to be complete, right? The whole point is eliminating the degrees of freedom that are not important. I think there is a big difference when you want to build a force field, and coarse graining force fields might also be an example in case in which these kind of problems matter a lot. And when you want to, you know, simplify the description of a complicated system where it's often possible to eliminate, I don't know, hydrogens or components that you kind of know cannot be important.
Yeah, so maybe along this line. So, you know, obviously people in the biophysics community talk a lot about the importance of water, right? And so to stability, conformational changes, dynamics, and all that. And so, and very often, you know, we want to, I guess, remove the solvent coordinates as much as possible, or represent them as compactly as possible. So, you know, what's your take on that? You know, how easy it is to formulate, solvent feature in the kind of problems that you guys deal with.
Yeah, that is actually a question that I have for actually Frank when he was talking about the Boltzmann generator. It makes me think about like how he represents the water degrees of freedom. It is very, very challenging. So then we currently do not include the water information. And then for cyclic peptides, then I think it can sometimes miss structural information because of the ring kind of structure. So sometimes you do see caged water molecules actually has a very long residence time interacting with backbone or side chain of the cyclic peptide. However, that kind of feature is going to be very difficult to incorporate in the model, and at least I haven't come up with a way that says that, hey, if you somehow have these structure, these amino acid in this pattern, you are going to have a caged water molecule, and that's going to make this structure super stable. So with that, I think that's still going to be difficult. Then it might be some sequences or structures that we'll actually miss on.
So we deal with waters in a relatively crude way, which is we don't account for every single water molecule at a time, but we look at solvation states of specific residues, which can be monitored. And so in our work, when we look at problems such as ligand dissociation, I don't think it's a good idea to go to implicit solvent. It's the waters, cage waters are always there. You're deeply shielded hydrogen bonds, and if you and when instead of just accounting for every, so you let go of the degen, you assume every water molecule is same, which is probably not true, at least in But if you can account for the solvation state for every residue, and that can be used as a feature. And this is kind of similar to how people such as Michele have looked at crystal nucleation, looking at coordination numbers. So that's the approach we take for waters, which is collective, but it's better than implicit solvent. I would say it's slower, though, of course.
Yeah, I think that this is actually one aspect in which there would be a lot to be gained by incorporating more of these systematic machine learning features, because you can go beyond coordination numbers that are basically just pair terms, but you start also getting in information about three-body terms that can describe in a more complete way the coordination state of a residue.
So I have a question for you, Michele, in this context, if I can.
Mm-hmm.
So you said, okay, we can go beyond pairs, we can go to three-body, but our common mentor, along with David Chandler and others, have papers on water, how it forms 10-membered rings and cages. You think we will be able to account for such very long-range correlations?
I actually, so in general, I think, so all the long-range part is a very interesting aspect. My personal, so this is now getting very personal, so we have a paper in which we look at zeolites. Zeolites, they are the same thing as water, tetrahedral network, very similar actually. And the zeolite chemists typically look at these structures in terms of rings and topology of rings. So this is actually something that I like a lot to do, and I think it's a very interesting application of machine learning. This is what I call a knockout model. So I don't know if you know what is a knockout mouse. You can basically buy for $10,000 a mouse without a gene. If they can make it viable, you can buy a mouse without the gene. And you can do something similar with machine learning models. So you can build machine learning models in which you artificially remove some information and you see if the machine learning model becomes crap. And actually, if you try and objectively look at the problem of predicting the properties of a zeolite, using information on the topology, it doesn't work. You can't predict anything using as input information the topology. So my personal impression is that, look, all these ring structures, that they are beautiful to look at. They might be also good to get some kind of heuristic understanding of what's going on, but I don't think that they are really very predictive. So you probably don't need to incorporate information on complicated network topologies, because I'm not sure that it actually matters.
Well, that's very interesting, and certainly, I don't know, makes life a little bit more promising, and so that I don't have to worry about five-body correlation when they develop models. All right. I think there are a couple of more questions for Yushan again. Okay, so let's see. Hi, Yushan. Is it possible in vivo your cyclic peptides will recapitulate the helical side chain configuration with higher probability by hydrogen bonds stabilization through helical interactions? Or do cyclic peptide not interact with helices through a stacking interaction?
So those sound like two separate questions. So then the first one related to if the cyclic peptide can be used to mimic a helix. I guess is a question. So then typically for mimicking a helix, people will do what you can call it cyclic peptide, but typically we call it stapled peptide. So then it's a helix, particularly a helix mimetic. So then it forms a helix with a side chain stapling, for example. And then for using cyclic peptides to mimic helix-related motifs, I think there are probably examples of mimicking a helix cap. So then like the cap at the very end of the helix, it actually kind of does just like look like like kind of a loop structure. Yeah, and then so regarding if cyclic peptides interact with helix or not, so I think it's possible that you can design cyclic peptides that will interact with helix. So I'm not saying it's impossible. So then, but then that will be more along the line of not structure-based design based on what you already know what to mimic, but based on this is a target I want to glue to, and then design something that will glue to it. So then I think that's even higher level therapeutic design. And so what we're doing right now is we know these two binds, and then if I want to bind this, mimic that. So that right now we are working on easier problem, but I think it's possible if we understand the cyclic peptide structures, and we also have a way to actually gauge the interaction affinity between a specific structure and the target that you want, then that could be achieved. So we can use the structures generated by our model and then do a screening or some kind of docking or free energy evaluation for it.
All right, very, very cool. So just one comment, I guess, from Vale Molinero. I'm sure many of you guys know her. Michele, we're working on zeolite nucleation, and you're right, you do not need further correlations to nucleate zeolite. The 10 or 12 member rings are very local and can be made with short range models. And so, yes, I guess very good to know.
Thank you. I was fearing that I had exposed myself a little bit too much. Thanks for covering for me.
That's great. That's great. Let's see. So let's have another question. Okay. Do the active learning techniques that we hear about with respect to automated experimental systems, like in the morning, have a place in MD simulations? More specifically, could these techniques be applied to predicting cyclic peptide conformations, in Yushan's case? And then also, would it be correct to think of a Pratyush's approach be considered a type of active learning workflow? So I guess we can hear from Yushan and Pratyush.
Yeah, I think it could be very interesting, but I do think that we will actually need to represent the amino acids using some feature vector rather than using like this is alanine or that is aspartate. We need to actually make it a numerical representation so that even potentially then you can move from, oh, I think if you want to stabilize this structure, you should increase the whatever property, then you go for the amino acid that trends along that direction. And then I think with this idea, you can probably go to experimentalists as I know that if you give me amino acid here that has this property, it will stabilize this structure. Do you have an artificial amino acid that you know about that will fit this field? So I think that is a very interesting idea.
Yeah, and I would say indeed what we are doing, this notion of iteration is a form of active learning, but I get confused sometimes with all these terminologies because one another term that comes up a lot is reinforcement learning. And only 3 days ago, there was a news article saying that DeepMind people are claiming that reinforcement learning is sufficient to get general AI, not just the type of function fitting AI we do, but really intelligent AI. So between active learning and if you think about metadynamics, that is reinforcement learning, right? You're reinforcing behavior that you don't want to repeat. So between reinforcement learning and just active learning, I get a bit confused which is which. But if you iterate, it's the same idea, using data to design new experiments, to get more data, to do new experiments. But going back to the first question David Coker asked, we have to be very careful that we are not just taking off in random directions and just doing random exploration. So yeah.
And I should add that active learning is fantastic when you want to build something like a interatomic potential, or in general a model of the of the properties of your system, because you can basically, as long as you can predict the uncertainty in your machine learning prediction, you can just run your simulation and stop if and only if it starts giving you results that are not reliable. And this is, to me, is the most pragmatic approach to building machine learning models. You just start from a guess, and then you do your thing, and you update the model whenever you need it.
That's certainly a wonderful flow. All right, let's see. More questions for you, Sean. Would dealing with other common macrocycle components such as peptides and methylation or non-natural amino acids also cause issues if the conformations related to those features aren't fully described by only the phi psi angles? I guess this is related to, you know, the expansion of feature space.
Right. That is certainly true. So then, for example, related to N-methylation, it will increase the likelihood of cis peptide bond formation. And then based on the limited cases that we have tested, the current simulation force field, I think if you give the right cis trans isomer for the whole cyclic peptide, it can predict the structure correctly. But then it's unable to predict if you're going to be a cis or trans peptide bond. So then meaning that complete de novo prediction of the structure for N-methylated cyclic peptides at the moment is difficult. So that's certainly a challenge. And then I think the force fields being generated typically assume that the amide bonds are in the trans conformation. So in general, what we see is the cis peptide bond formation is greatly disfavored. So then if you, in your meta dynamics, allow the omega angle to change, what we saw so far is that the trans conformation is always preferred. If we want to use our method from targeting the couple dihedral angles for other systems, I do think that it's probably a common feature for small size rings that you will see this kind of couple motion because the ring strain will prevent you from doing a single dihedral rotation because you're going to break the ring. And then so the two dihedral angle motion is a way, one of it is called a crankshaft motion. So then it kind of holds the two ends kind of not change, and the middle actually can twist, right? So then, so I would expect that this might be an effective collective variable for small size macrocycles. For large size macrocycles, then I think we are going to run into a challenge of the sampling space. The configuration space is so large that it will likely still take a very long time for us to converge. So then another observation is your simulation typically converge faster if your peptide is well behaved. So then if this system has multiple low-lying free energy conformations that are very comparable, then it's very difficult to gauge those small differences in free energy minima. However, if there's a clear winner, then it tends to go there very quickly. And then so in this case, what I will probably be hoping to do is that the sequence space we're interested in will be the sequences that are well structured or more or less well structured, right? So then we don't want the intrinsically disordered peptide. We want this conformation. We want it to have significant population. So what we might end up doing is we won't be able to predict precisely what the structural ensembles for all peptides look like, but I might be able to tell you which sequences are likely to be well structured and then what the structures are.
Mm-hmm. And then likely they're most effective, right? And so for the specific application, yeah. All right, so I better get to this particular question because I think it's from a former graduate of our department, David Stelter, who worked with Professor Tom Keyes. Anyway, so he said, do you think that an additivity / feature construction based method could be applied to finite rate chemical kinetics and basically looking at, pairs of molecules and inferring their cross section, for example. Kinetics always, fascinating.
I think in principle, yes. This actually touches a little upon a more general issue, which is can you use the kind of featureizations that I talked about to describe chemical reactions or a sort of macroscopic properties of molecules? Let's say that you want to predict if a molecule is toxic. Now, strictly speaking, this is not a property of a specific conformer of that molecule. And so the kind of representations that I talked about that are really designed to precisely link one and one only distribution of the atoms to a very precise quantum mechanical property are not necessarily the best for this purpose. With that said, I think that the structure of additivity properly expanded to describe in a more with a better fit with the problem, this issue of assigning properties not to a specific conformer, but to an ensemble of configurations. I think that yes, it could be. And there is actually a very recent article by Clémence Corminboeuf on chemical science in which they use similar features to predict the enantioselectivity of catalysts. And roughly speaking, it works by assigning features not to molecules, but to pairs of reactants, very hand-wavingly.
So
Any other
chemical kinetics opens up I think, a deeper question. What does it mean to get a rate constant? We have training database, which comes from structures in their ground state. Kinetics is really one, even if you believe in transition state or not, but you do agree there is some activated state that you have to go through. States that have not been input in the training database. So if you have indeed learned the system's Hamiltonian so well that you can go and interpolate between ground state structures and look at the transition state, we know it would work at 0 temperature. We would get saddle points and all these things. But how do we take care of entropy? How do we take care of entropy at the transition state? So I have been battling kinetics for a while, and I think it's very hard to get it out of machine learning, actually to predict kinetics. To capture kinetics from reactions that you have seen is one thing, but in general, there is a question, how much of interpolation are we doing with all of this? Are we doing any extrapolation to begin with? And I think for kinetics, we have to do extrapolation, which I don't know if it's possible. So I think there is more careful, which goes back to the poll that I gave. It's partition function that is common, but not necessarily kinetic properties. So I think that's going to be hard. It's not going to be that easy.
Well, I agree. I remember having a discussion with Vijay about this, and then I think Vijay's perspective is if you simulate so many molecules and then you can get the ground state Hamiltonian correctly, then there's a hope that maybe you can also describe these transition states well. However, to actually use kinetics as a property that you use to tune your force field is very challenging, because it's going to be such a time-consuming simulation to do, and then you need to iterate and then tune your parameters to see what kind of kinetics you're getting out, and then you can also have different kind of pathways or mechanisms that give you the same rate constant. Then how do you know that if you are actually tune the parameters right to get these very few numbers of rate constants? So but it is certainly a very interesting and then even more challenging goals that computer chemists can work on.
Actually, following up on these comments, I think one aspect that is certainly true is the fact that part of the reason why machine learning has been so successful is that a large fraction of the chemistry that we care about is relatively low energy. So we have this data set of random CH4 configurations. So basically a carbon and 4 hydrogen atoms distributed randomly in a sphere of 3.5 angstrom in radius. And actually you need of the order of 3 million configurations to predict the energies of these set of configurations with 1 kilocalorie per mole accuracy. So if you really have, but I do think that transition states are still not, they are just still a relatively small portion of phase space. So there is some hope.
Yeah, I think this is a great way to quote unquote wrap up this particular question. It's not an easy task, but still hopeful. So we have maybe 2 more minutes for a last question, which I think is almost like perfect for concluding this part. And so it's targeting Pratyush, but I'm sure others would have comments as well. Basically, we have seen how machine learning is aiding computational simulation of molecular systems as statistical physics, lead to new insights or innovations in machine learning methods.
I can give a quick answer. I can just refer to 2 people, one at Google Research. His name is Alex Elemi, and I recommend reading his papers, especially deep variation information bottleneck. He's trained as a physicist with Jim State now. If you go and ask him, he would not have done any of these methods. He has a wonderful paper called Thermodynamics of Learning, which will answer your question. And also checking papers of some of the people out of DeepMind at Google, and you will see there also they have how free energy perturbation can lead to machine learning methods. So definitely it's influencing both ways.
I will add another name, Lenka Zdeborova. So she has been doing very nice work on studying the learning of neural networks as a phase transition. So really statistical physics applied to deep learning.
All right, and so I think this is a perfect time to conclude this session. So again, I really want to thank all the speakers, Pratyush, Michele, and Yushan for their wonderful talks. Again, really, shows how machine learning can be integrated, into all kinds of interesting applications, and there's still a lot of room for creativity and innovation, and so certainly integration between machine learning, data science, and physical sciences, right? Statistical mechanics in particular. So now I should also thank the audience for participating and giving us all those interesting questions that led to all those fascinating discussions. So I guess now I'm gonna hand it back to Emily and Aaron for concluding remarks.
Thank you so much. Thank you to the whole panel for session 3 and all the sessions. It was wonderful, wonderful discussions. We're just going to take 10 minutes or less to kind of wrap things up, discuss some of the notes that we've taken and things that we've kind of highlighted out of the sessions, and also give some very big thank yous to everybody that's helped make this day possible. So let's start by talking about the sessions. And Aaron, just jump in. Aaron and I have tried to take some notes on what we thought were some of the kind of important things we heard out of the sessions. Is definitely not inclusive of everything that was discussed, but we'd kind of like to discuss them here as some of the challenges and other things we've seen. Aaron, do you want to take session 1?
As going into it, I introduced the session as a lot of discussion around the challenges of generating robust and useful data for predictive and machine learning applications. And ultimately, I think those are the exact, largely, a lot of the points were right in line with that. It certainly was mentioned multiple times that capturing the large number of discrete and continuous variables within a data set is challenging, but even more so in chemical reactions, because individually one of those could absolutely dictate the outcome of the reaction, and it's not always clear how that connection is being made. Reproducibility in chemistry is challenging. So this is a recurring theme, one that we hear not only in symposia like this one, but I think this is one that we hear in chemical sciences over and over again for a long time. And really developing technologies. One of the things that was a thematic through all 3 of the talks was utilizing flow chemistry and oftentimes robotics to help facilitate reproducibility and robustness of data. And ultimately, as I think is similar to a lot of the discussions that came up later, when we were thinking about the interaction of molecules, and in particular, reactions in which there are multiple mechanistic possibilities. This only further complicates all these other issues. And so I think that's one of the reasons, and again, I think something that came up in multiple ways, one of the reasons why chemical sciences is particularly challenging area to predict. Certainly, again, it's something we've heard for many years in various contexts, is that the lack of negative data in chemical literature is a major issue. And I think the speakers did a great job of discussing how they're trying to overcome this, why that may be. Connor's efforts, such as the Open Reaction Database, are great starts to addressing this. But I do think that it's so reoccurring. This idea that negative data is lacking is something that chemists as a whole should address. And ultimately, analytical bottlenecks when it comes to actually generating data in a timely and cost-efficient manner. Analytical techniques are really a major bottleneck, and this is something that, while this was not, I guess, discreetly discussed in these talks, this was again a recurring sort of theme, and something that hopefully in the coming years we'll see addressed. So with that, I'll pass it on.
Yeah. So session 2, focusing on machine learning applications. I think there's a lot of things we pulled out of this. First one being the critical and challenging aspects of modeling or mapping molecular structures and adhering to the physical laws when you're trying to provide accurate descriptions using machine learning applications. I found it very interesting, the conversation around the ideas of deep learning as we're moving more into that kind of area, and its flexibility and high potential for these complex systems. But the question of, is it always necessary or appropriate, as opposed to, as they said, the shallow learning applications, and that trade-off that we heard, I think, multiple times throughout multiple talks about the trade-offs between complexity, computational cost, availability of data. What I really pulled out of that was just that you have to be flexible in the methods you want to apply and really think about what's appropriate for the problem that you have at hand. And then something I know very well, and I think it's really key, is the idea of bringing together disparate disciplines. We heard a lot of discussion of that, and I think today is a really great example of trying to bring these communities together. We all speak very different languages. I am an engineer, not even a chemist. And I know from working with chemists, chemical engineers, computer scientists, et cetera, that it is very critical to try and develop those relationships if we really want to move forward these really cross-disciplinary and convergent areas. And I like some of the aspects I heard from the speakers here about developing or helping to define overlapping interests and making sure you are both kind of invested in the problem, and trying to show what you can do with the other in the domain, as it were, or with machine learning, and how it applies to the problems of interest to the other people. And I think this last bullet is really critical, and it's the idea of building long-term collaborations where you have both parties really invested in understanding each other. I think that's the only way we can really move forward all these ideas we've been talking about today, is to build these communities and this collaboration. And I think, I forget which speaker it was, I'm sorry, but there was some comment of, don't just do it for a paper, do it to really move the research forward. And I think that's really a key thing. And I think days like today are going to help do that and build that community. And then the last session, this one got the least attention because it was right before us. So there's probably a lot of typos, et cetera, in here, but a very good discussion for session 3, looking at the computational aspects. I think there's some great discussion about how data science is developing more flexible atomic scale models, but also this idea of, I think where we really need to go is making sure we have physics informed machine learning or in incorporation with the data sciences, and really moving that forward, so that we're making sure we're really getting an integration going both ways between the physics and the data sciences. I heard lots of challenges kind of focused around, and I know it very well, the idea of computational expense, from trying to generate the data to use in machine learning, to the fact that we can't get around the fact that time is inherently sequential and it's very difficult to parallelize. And then an always critical area, the ideas of convergence and accuracy, verification and validation, and really proving that what we're doing is accurately reflecting physics. It's often time consuming and difficult to prove. And then I heard more about the ideas of going beyond kind of pair-pair interactions and coordination numbers, and the question of, is it really worth the cost to get those longer range correlations? And again, is it really needed? So that idea of thinking about the problem you're dealing with and what's really necessary for it. And then that final discussion around kinetics, I think it's a great discussion. Can we really use these featurizations to describe the chemical reactions or the macroscopic properties? And it is definitely hard to do for machine learning, especially this idea of extrapolation. I'm talking about that a lot within my group right now. And so just a lot of great challenges, I gotta say, were brought up in all of these sessions, and a lot of great ideas. And I hope you guys really took something away from it all. But from what Aaron and I heard, those were just a couple of our thoughts on the sessions. And so thank you again to all the speakers. And I wanna say a more formal thank you so we don't hopefully forget anyone. Aaron, jump in when I do forget somebody. Obviously, the Focus Research Program community that Hariri's been helping to coordinate for us. Thank you to all the faculty, the graduate students. I hope you guys went to the poster sessions, all the hard work that's going on. I think we are developing a really great community here at BU. Obviously, thank you to everybody that participated today. The panel moderators, Malika, Brian, and QC, thank you for doing that. And the panel introducers too, as well, Kostas and John and Aaron. Huge thank you to Hariri Institute for Computing. Without them, this would not be possible. They provided us with seed funding for the last year to start working together. Obviously, they provided all of the technical and organizational backup for this symposium today, especially Catherine and Gina, who were amazing and really made this possible. And then thank you to all the colleges and departments at BU that co-sponsored the event. We really appreciate their support with this. And then a couple of last words and we'll be out of here. First off, please join us again. The Hariri Institute has another focused research program ongoing. It's about leveraging AI to examine disparities and bias in healthcare. And they will be holding a symposium, something similar to this. I hear it might actually be in person. We'll see, in October. So hopefully they will be able to do that in person. And I hope you'll join us if you're able to hear what they've been working on. And then I'll close it out by saying, please, stay in touch with us at the Hariri Institute and at Boston University. We had a lot of faculty involved in this, and I'm sure they'd love to hear from you. And you can find out more on all these fancy social medias of Twitter and Facebook and YouTube. And most importantly, as we've said throughout the day, the whole symposium has been recorded, and it'll take us a little while to post-process it, but by the end of the week, we should have it all up on our YouTube channel. So the Hariri Institute for Computing at Boston University we'll be hosting that on our YouTube channel. And I think with that, we are exactly on time to end. Aaron, have I forgotten anything?
You have not. Thank you, Emily. And
yes.
Another thank you to all the participants.
Yes, thank you to the audience for joining us and helping the conversation go along. And I hope you enjoy the rest of your day.